everyone. It's Andrea here from Standard Imaging, along with my colleague Shannon.
Hello.
We are here today to welcome you to our second QA Essentials webinar series. We have some more coming up after this one, of course. Like I said, I'm Andrea. I'm one of our product managers here at Standard Imaging. One of the product portfolios under my purview happens to be stereotactic phantoms, which will be part of our conversation today. And as I said, introduce my dear colleague, Shannon Holmes, a PhD physicist. She is definitely a subject matter expert for me at the company because I am not a physicist. But not only is she one of our in-house physics experts, she's also a product manager for our X-ray line of detectors and our electrometers.
Thank you, Andrea. It's always fun to chat with you about these things.
Yeah, I always learn something when I talk to Shannon, so hopefully you all do too. So we have a bit of housekeeping just before we get started. Just to remind everyone that you are in listen-only mode. Please use the dialog box to type in any questions you might have during our conversation today. We might be able to squeeze them in while we're having the conversation. Otherwise, we'll wait till the end and make sure we check on them then. Today's webinar is being recorded, of course, and it will be available on our website shortly for your pleasure. So let's get started with the QA essentials for stereotactic. Let's get it going. So I'd like to start with Shannon, asking a question to Shannon. If you were to start an SBRT or an SRS program, or you're a physicist walking into your new clinic where you've inherited a fledgling stereotactic program, what sort of things would you consider as essential for a stereotactic program?
That's an excellent question to start with. I usually would walk into a situation like that and start by thinking about, what do I have to do? What are the tasks that I need to complete? Because that will help me choose the tools that I need in order to complete those tasks. So for an SRS or SBRT program, first and foremost is calibration and commissioning of usually small fields for these treatments. And then next would be making sure that your entire process of your treatment, from scanning and planning, sending plans to the LINAC, and then treating your patient, all of that fits together to give you the accuracy that you're looking for in your treatments. And then last but not least, making sure that once you set it up and you're confident it's accurate from the beginning, making sure that you monitor that over time in order to ensure that you continue to treat patients with that same accuracy that you established at the beginning of your program.
Okay, well, there's a lot to cover then, I see.
Yes.
So now that we know sort of what your essential tasks are, really what tools, what's going to help you figure out what are the right tools for the job or the jobs?
Yeah, so under calibration and commissioning, you need a good detector first and foremost, an electrometer to work with that, and usually a 1D water phantom, some way that you can actually do an in-water measurement for output. That for that second task, the process steps, you need an appropriate phantom for end-to-end testing that matches the sort of treatments that you're going to be providing to your patients. And then that longevity monitoring over time, you'll need Winston LUTs. That's something that's required. Potentially some software tools, and then of course those calibration tools come back into play again for periodic monitoring of your beam output. So if we start with those detectors, when you're choosing detectors for small field calibrations, you really should keep in mind that all detectors have both strengths and limitations. So in my opinion, it really is best to have some complementary tools. So something like our 826 micropoint chamber gets you an ionization chamber, so you can have NIS traceability for dose outputs for these very small fields with a reference class chamber that is very small. You can use it for small field scanning, but we also have tools like our diodes, the D1H and the D1V. They're similar diodes. They're identical diodes. It's different housing in terms of the orientation that you see there. It's a 1 millimeter diameter spot size there, the region that's actually the active integrated region for those diodes. They're unshielded, so they work for both photons and electrons quite well. I should have mentioned the A26 has about a 4.3 millimeter spot size, so it is very small as far as ionization chambers go. The W2 scintillator is also a very popular item for small fields because the scintillator is water equivalent. And so the W2 has a beam quality correction factor, KQ, of 1 unity. So you don't have to worry about the change in your energy spectrum as you get down to these small fields with things like source occlusion and the change in the penumbra due to the energy changes, that sort of thing, or lateral scatter problems, because it really truly is water equivalent. So that gives you, sorry, go ahead.
No, I was just going to say that's great, because I bet people or physicists really appreciate having a KQ of 1. No correction factors. Yay!
Yes.
Something to make their day.
No digging through publications to figure out where the correction factor is for this particular detector or that particular detector. But it is, I mean, in terms of those strengths and limitations, ion chambers have a low density region. They have an air cavity. Diodes have a high density region. And so there's a balance there between the two of them in terms of the correction factors and the energy dependencies when you get into these crazy small fields. But it is certainly nice to have something that doesn't require quite that much complexity in terms of the changing of the fields and the changing of the energies.
Yeah, and 1 millimeter resolution is just phenomenal. So that's great.
It's great. Yeah.
Well, so that kind of gives overview of our small field detectors. Thank you so much for that. But they don't work alone.
Right.
So the next step is electrometers. So yeah. What are sort of the important considerations in selecting an electrometer?
Well, you definitely want a high quality electrometer. You want one that's stable and one that's calibratable, really, so that you can get high quality measurements over and over and over throughout the lifetime of your electrometer. It's important with these small field measurements to make sure that you're matching the range of the electrometers, the capabilities of the electrometers, with the magnitude of the signal that your detector will generate. Ion chambers in particular are the signal generated by an ion chamber is directly proportional to its air volume, its active region. And so as you go down into these small ionization chambers, you get much less charge. So you need an electrometer that can handle those low ranges. Another thing to consider is warm-up time. Are you the sort of person who leaves your electrometer plugged in all the time and can use it then whenever you feel like it? Or are you the sort of person who sets it on the shelf and then, oh dear, last minute, I need to run in and make a measurement because suddenly I have an opening on the LINAC and I've got something I need to measure, and that short warm-up time is really critical for you.
Yeah, that would be me. I'd be the last minute spur of the moment person.
Yeah, I tend to just leave it plugged in. That's me. I just leave it plugged in.
Yeah, see, but that's you and me, yin and yang.
There you go.
That's why we collaborate so well together.
we make a good team. I agree. So in terms of our electrometers, the Max 4000 Plus, which is at the top of this slide, is our kind of the workhorse. It's the robust single channel electrometer, reference grade, used for countless clinics. I don't know how many of these we have around the world, but this is a highly popular electrometer. The Supermax is our top-end electrometer. It does have a touch screen. It has dual channels with independent bias, and it has that short warm-up. It also has a little bit better resolution at the low signal end of the range. But the last thing I would want to point out, and I guess I have a note on this slide as well, the last thing I would want to point out about electrometers is pick something with a good warranty, because I really feel that with electrometers, as with a number of other pieces of equipment, the warranty that's offered with it is a good indicator about how dependable you can expect it to be. And we stand behind our electrometers. They have the industry-leading 5-year warranty. I think it says a lot about the quality of the electrometers, that we expect them to last a long time. You should be able to expect them to last a long time too.
That makes a lot of sense, and I completely agree. If you're going to invest, let's protect that investment, and we're here for you for that. That's great.
Mm-hmm.
So, of course, I have another question because I'm the queen of questions here. So can either of these electrometers be used with, like a 1D scanning system?
Yes, yep, they both can. They both work just fine with our DoseView 1D tank. Really, the question on which electrometer you choose in terms of the water tank is whether you feel you need to have a reference detector. You can get high quality measurements from our 1D water tank if you're scanning just with that one chamber, get your depth dose scan, not have to process it in order to make it smooth and all of that. You can get a good set of measurements just with that single chamber. But if you're the sort of person who really feels that they need to have a reference chamber as well, then you would need the dual channel electrometer in order to enable both the field chamber and the reference chamber.
Okay, so what I hear from you saying that is that we build such a robust system that you get excellent quality scan data from the get-go. So your raw data with a single channel should suffice. And
Yes.
And for many, many people, it does.
Yes, that's absolutely correct.
I just want to make sure I got it right.
Yep. Yep. So the water tank works with our DoseView software. It has a very high accuracy for positioning of the detector within the tank, and there are a couple of leveling adjustment screws on the back end here. Let me point out on the slide. Leveling adjustment screws that help you adjust if there's a slight tilt skew that you need to account for when you're setting this up.
Well, okay, so I have a question then. So you said that with software, so do you need software, or can you tell me something about our software? Would go with DoseView?
Yeah, you don't have to use the software. You could certainly drive the chamber to the depth, align a surface, set your origin, drive it to the depth that you want to make a single measurement in, and make a measurement with the electrometer as just kind of a standalone system. You don't have to work through the software. However, if you want to scan a % depth ionization curve and you want to convert that to dose, and then figure out where your, say for electron beams, find your DREF, then put your chamber at the reference depth, and then make your measurements for TG51 or TRS 398. You can do all that through the software interface. And the DoseView will accommodate, it'll accommodate the W2, it will accommodate those micro chambers, pancake chambers, the parallel plate chambers, or standard farmer chambers, really all the detectors that you need for the TG51 or TRS 398 beam calibration measurements can be accommodated in the 1D tank.
See, there you go. You proved just how well we work together because you anticipated my next question, which was the detectors you mentioned, do they work? Obviously they do. Well, that is great.
Yep.
Okay. So what have we got next?
Well, I did want to point out, I guess, that our DoseView software is the same for the 1D tank and our 3D tank. So it's really the same interface. You're pulling up the same software, and you're either saying I'm working with my 1D tank or I'm working with my 3D tank. So it's the same piece of it's the same interface. It's the same piece of software that you would use for both tanks.
Great. That makes customers' lives easier, I would anticipate.
Definitely. Yep. And we've been told our DoseView software is pretty much the easiest, most straightforward, most intuitive software available for 3D tanks, which is praise that our software development team really enjoys hearing when that comes straight from the field. But with the DoseView 1D side of the software, we did add that single point measurement functionality, so you can do your PDI, convert to PDD, go to that reference depth, and make single measurements. Or if you just want to use the interface to make individual measurements at various depths, you certainly can do that. Plus, if you do have the 3D tank, you don't have to relearn the software. every year when you pull it out for annuals, or if you have to pull it out for commissioning, even though it's easy to relearn, renavigate, if you're using it more on a monthly basis, it's completely familiar to you when you get to time for annuals or for commissioning.
Yeah, that's fair. And I will say, again, non-physicist speaking here, I had to assist the dose view product manager on site, and he was, doing setup things and running the show, but he's like, hey, Andrea, we need another scan queue. Hey, Andrea, we need another this. Hey, and I'm like, okay. And it was incredibly fast to do. I was amazed because I'm like, please don't, this is new to me. Make it simple. And it was dead easy.
Excellent.
So that was pretty exciting.
Excellent. Oh, I had a question that I missed. Is the diode shielded or unshielded? It is unshielded. So it is good for both photons and electrons. But back to our slides at hand, I guess we're going back to our task list here. So now I have a question for you. If we're looking at end-to-end phantoms, how do you know what is an appropriate phantom?
Well, that rather depends on whether you're talking about SRS or SBRT, right?
True. Yeah.
Fingers crossed, you're not walking into a situation where the doctors immediately want to do multiple metastases with a single isocenter in the brain. No, please don't do that. I don't have any brain cells to spare. Let's make sure we're doing QA right and using the right tools for the QA, right?
Mm-hmm.
So since they don't usually start with those multiple metastases in the brain, I've often asked customers, if you're starting a program, what are you starting with? And very often it's SBRT, and it's often SBRT lung.
Sure.
That seems to be a real common one. So let's go ahead and start by talking about SBRT and what phantoms we would have available for that. There are certainly things that you need to consider, right? How realistic does the phantom need to be in your mind? Does it need, to be large and shaped like the thorax? Does it need to include organic shapes, such as a kidney or a lung shape? Or on the other hand, do you prioritize the heterogeneities? And if you are thinking heterogeneities, do you have a certain number that should be included in the phantom in mind? Do the shapes matter? Does the position within the phantom of those heterogeneities matter? Another thing to consider is whether motion management or gating, is important. Are you using those systems? And if so, what QA might you need for that? And in which case, if you're doing gating, here we show at the bottom center our respiratory gating platform. Either of the phantoms shown here would easily fit on their platform for you to be able to do your motion management. And then, of course, unfortunately, none of us have unlimited expenses, so we have to consider the budget. It'll impact what you're ultimately going to be able to acquire. There are price differences between our thorax phantom and our stereotactic dose verification phantom, but at least we have options for you in a couple of different price ranges, which is a good thing.
Definitely.
So,
go ahead.
Go ahead. I'm sorry.
I was just going to say there's a lot of stuff on this slide. I know we have quite an array of phantoms. I was wondering if you could point out the key differences between some of these SI phantom solutions that would help influence someone's choice of which phantom would be best for them.
So it really does. Well, I'll do my best here.
That's all we can ask of anybody, right?
Yeah, so I will admit that our thorax phantom is newer to me because it's one of those items that we got in our acquisition of IMT.
Mm-hmm.
So I'm learning about this one, but it's really nice. We have changed it up. It is now going to be made of our virtual water material.
Oh, that's great.
It is a thorax, shape and size. So again, if that matters to you, that's what's going to matter. It's made in 4 sections. And as you can see here, sort of in the light blue version, the variety of items embedded within it. So if anthropomorphism on some level is important to you, you get that with the thorax phantom. You have ribs, for example, vertebrae and a spinal cord. for localization and for different, and they are different densities, of course. We do have a triangular shape, which is of course not a natural human organ. Sometimes you need simplicity, but we also have this kidney shape here as well, where you can place the ion chamber or other detector both within the target and within the target margin, or the margin of the target, which is, I think, a differentiator compared to our stereotactic dose verification phantom and probably other phantoms on the market as well. So again, we have a variety of densities. There's a variety of resolution markers within this phantom. It's quite a complex phantom for you to accomplish a lot of QA. And again, I'm not as familiar with it, I'm learning, but there's a whole bunch of CT QA that you can do, as well as your SBRT QA with this phantom. On the other hand, if the form doesn't need to be pseudo-anthropomorphic for you, you're used to sort of the simple older days of IMRT phantoms, then you can consider our stereotactic dose verification phantom with the heterogeneity inserts. The heterogeneity insert here is the gray lung slab material that can be added to the phantom.
This one's more budget friendly too, right, Andrea?
It is definitely more budget friendly. There's definitely the differentiator here. But you have the lung material, and then the chamber plug acts as the tumor, different density tumor, so that you can do your Monte Carlo type, calculations. We also offer the QA slab, which you can see with the dark shapes here are tight and difficult target and avoidance structure configurations to really test the treatment planning system's reconstruction capabilities. How well to really target on those, and can you, plan to it and treat to it, and then verify. Both phantoms use detectors and film for the verification in the workflow that you would have. We do include also in this QA slab a variety of different densities. So again, they're not an organic shape, but they're there. You have the air pocket, the blue water, so water equivalent, and you have adipose fat, trabecular, and cortical bone. So you can do that spot check of your CT densities as part of your QA and your treatment planning system. The other thing that I will point out, because again, we mentioned sort of if you're using SGRT, the virtual water here of the RX Phantom is visible on all of the systems that are out there. We have tested, I literally hand carried, we were like the three kings of Orient are, as one person, I carried the SDVP, somebody carried our Mimi Phantom, which is White Delrin, and then we also took over the Max, another of our Max Phantoms made a virtual water, took it to Vision RT, took it to Variant for Identify, took it to CRAD for their system. And if you have Vision RT, then blue water does not flush. Straight up honest about that.
Yep.
So it is a consideration of, consider your materials and the texture and those shapes, whether they're going to actually be traceable, trackable on an SGRT system. So I think that's all I have to say about those.
So, okay. So what about SRS then? What additional priorities might we need to consider for SRS?
So as our phantoms point out here, anthropomorphism comes into play. Again, our Lucy Phantom is not anthropomorphic. How realistic does it need to be? Is pseudo-anthropomorphism enough, or do you need the full skull and full brain cavity, that type of thing? Again, your heterogeneities, are they embedded? What are they made of? How many of them are there? What shapes they have? Maybe that matters. That's consideration. Again, imaging modality compatibility. And you probably also really want to think about what type of Linux am I using. So what I mean by that is, we have an MR visible phantom. So if you have a Unity or ViewRay, that might matter. CyberKnife might something different. You know, you kind of want to take a look at form, function, what's going to work well for me in those. And of course, your budget comes into play.
Always,
Again,
always.
unfortunately, we all live with budgets in our personal lives and certainly in hospitals and clinics. So that is going to be into your decisions as well.
Yeah, absolutely.
So now I'm going to go sort of over the three that we have in our Lucy family of phantoms. Long time been the gold standard for stereotactic QA is our Lucy 3D QA Phantom. It is the most versatile in the fact that we have had it for such a long time that we've developed a lot of different inserts that can be used in the Lucy Phantom. It also means that budget-wise, it's not only tailored to what inserts you need specifically, but we can tailor it to fit different budgets. Of course, you know, the more inserts you choose select, the more expensive it tends to be, but there are things that we can work with you to say, well, you can swap this for this, or you can do this, and you're still going to get an absolutely excellent Phantom and be able to do your end-to-end QA, but we can go through those considerations with you. So price range is versatile to a degree with our Lucy Phantom. One of the things that I really like about our Phantom is that we offer irregular shapes that you see both for CT and MR. And that allows you to be able to do a bit of basic deformable registration QA. Because the Phantom is on a stand that is rotatable 360 degrees, you can shift that shape both position-wise, and it changes its shape if you rotate the Phantom 90 degrees to be able to do some of that deformable registration QA. One thing I didn't put on here that I'd also mention is that the precision of Lucy comes from the fact that we lock things into place. Yes, we do use tools to do that, but for example, here on the front of the sphere, you see the locking mechanism for a detector. And that means if you rotate your Phantom or if you move your Phantom, you're not going to have to worry about your detector sliding out of position and creating an air gap exactly where you want to take your measurement. So those things are really important and add to the features of Lucy.
There's nothing more frustrating than trying to do those measurements and having things come out not where you want them to be and ultimately realizing it's because you didn't put the detector in all the way.
Yeah, very frustrating. So I do want to point out what may be considered disadvantages. It again depends on your priorities. It's obviously not anthropomorphic, right? But again, you see you have those target and avoidance structure shapes that you can use. Again, we have a CT density, so you can spot check your CT densities. That's really good. And speaking of SGRT, Lucy is translucent and it is shiny, which just are diametrically opposed to SGRT systems. So it is not a good one if you're trying to do any SGRT QA.
We did have a question about MR compatibility, so I wanted to point out with the Lucy that there are MR inserts. You can do your end-to-end testing if it includes MR imaging. There's a distortion grid down here, so there are some options for Lucy to make sure that you include MR in that end-to-end testing.
Absolutely, yes. And Lucy's all about the known shapes and geometries, right? So your MR inserts, do they line up? You know, when you're doing your image fusion, do you see the 5 millimeter center fiducial to center fiducial on those? Those known distances and geometries are super important in that imaging and treatment flow.
Yep, and matching the target on the CT with the target on the MR, that should be the same volume, should be the same size for the Phantom plan.
Absolutely. The next one I'd like to talk about in our Lucy family is the Lucy EA family. And this one is growing on me more and more. Again, this is one that came over with the IMT portfolio. We are now making it in our virtual water. The EA stands for easy access, and that really is true. So if you use an open mass system, you just have to lift off the face and you swap in your other insert. Nothing else needs to change in your setup. It's advantage, so we have some anthropomorphic features in it. We call it pseudo-anthropomorphic phantom. You see the vertebrae and the spinal cord. You see portions of the skull, and that we have air cavities in here. It is a nice, simple phantom to use because we have very limited inserts for this one, which also makes it very budget friendly.
That's a good thing.
So the included inserts are the one on the right here, which is our multi-target 3D film insert. So it's got 4 targets in it, plus a ventricle, low density ventricle avoidance structure. So that's really good. And then we also include the multiple target Winston-Lutz insert. And one of the things we did when we acquired it, upgrade a little bit, is we've added markings on the outside of the phantom, again, so you have your single phantom setup, so you could do your couch QA, the positioning repositioning test, using both the central fiducial in the multi-target Winston-Lutz insert and the 1 cm offset one. So that's a nice thing. You can, and then you can sort of see the offset mark here on the faceplate, but it's on 3 sides of the phantom. So that's a really nice thing. And then, well, as far as disadvantages, because we kind of are doing the pros and cons of everything here, this phantom is not MR compatible, in the sense that it is MR safe, but it's not going to do you any good because we do not have any signal generating components to this phantom. So if you're only CTing your customers, or your patients, it's a good choice. We do not currently have any MR inserts for this. The other item, which may detract for some people, like it for other reasons, is that the ion chamber placement is not at isocenter and it is extracranial, so it's not even in the brain cavity. Now, for those who are doing those multi-target single isocenter, it is a real push of the treatment planning system to have that extracranial target. So it has, it can be an advantage to have it there, but there are a lot of people who are used to having their point measurement at their isocenter, and in this case, you can't do that with this phantom. One thing we did do to help is when we acquired it, the solid plug was just a solid plug. We have now added a target at the same place as the centroid of your collecting volume of your detector, so that you have something that you can plan, scan, treat, and verify on with your point dose there. So I think that covers what we offer with the Lucy EA, and again, it is budget friendly. Last in our Lucy Phantom is the Lucy MR, and as the name indicates, this is a great phantom if you have a ViewRay or an Elekta Unity. It is also great if you're treating your patients, imaging them with MR as well as CT, because it is visible on CT. Just because it's called MR doesn't mean it's the only thing it can do. It is meant for CT and MR, and what makes it truly unique on the market is that the entire phantom, not just a few inserts like Lucy, the entire phantom is MR visible. So that is quite unique and great. And the other thing is that it is fully anthropomorphic. As you can see in the split image here, it's got the full skull. It is split horizontally, so you've got posterior and anterior sections of the phantom. And for this one, we provide an orthogonal film insert, both a posterior and anterior one that has a target. The posterior one is a smaller target than the anterior one. We also include the Winston-Lutz insert, which has a center, center BB and 1 offset fiducial for you to be able to do QA for that. We do offer a couple of other options with this. We have a 3D film stack insert, which I believe accommodates 9 films.
I think that's right. Yep.
I think that's right. And then we also have the nano DAF insert. If you're using nano dots, that's certainly something you can do. And again, throughout the volume, not just on a single plane, you can place those nano dots. So I think that really covers the advantages of this due to its the way this is manufactured, which allows us to have the full anthropomorphism, means that you do need to be aware that this is a longer lead time phantom. So when you're budgeting and planning and when is my system going to arrive, you really do want to consider the time frame for delivery and to ensure that you have everything you need from the get-go. The other thing is, while the ion chamber placement is located inside the brain cavity, again, it is not within a target. So a consideration that you're going to want to look at. And the one thing that may be a detractor, like the Lucy, we have an MR grid for distortion. We do not currently have a distortion grid for the Lucy MR. I'm sure that's going to be a question people come up with, so we'll be investigating it, but I haven't done anything yet, so it ain't coming tomorrow.
there was a comment from the audience actually that ion chamber placement off axis may actually help to measure out of field dose because that's also an important thing to track when you're talking about SRS. What is the rest of the brain getting? What are these other sensitive structures getting? So that's a very good point as well.
Yeah. So again, it's a pro and con. Some people like it, some people don't. I just, you know, you have to have a few disadvantages and with your advantages. You got, you know, it's
you
something
can't
that
please everybody though.
Yeah, right. And it's something that people have asked about, so I figured something I should anticipate the question and answer it.
Yep. But I mean, that's why we have options too, so that if one thing is much more important to you than this other thing, then hopefully we have a solution to offer.
Yeah, and I think it's great that we have a family of, you know, really head neck phantoms to select from. That is very unusual in our industry. It sort of actually speaks to something that has always impressed me, because remember, I've been in standard imaging for 18 years and seen the development of various inserts for Lucy and stuff, is that trying to meet the needs as they grow in the market, you know, has really been a focus, and SI's focus on stereotactic has been really quite incredible.
Yeah, definitely. There's quite an array.
Yeah.
So before we run out of time, I don't want to take too much of everybody's time. Maybe we'll switch to that last section of monitoring over time, how we make sure that we can continue to deliver accurate SRS and SBRT treatments. So
yes,
if you're
do.
I'm glad you agree. If you're treating with stereotactic radiosurgery, you need to perform a Winston-Lutz test every day. So you need something like a Winston-Lutz pointer. Ours actually has a 3D micrometer stage, so you can, if you do your Winston-Lutz and you say, well, it's offset by a certain amount, you can make those fine adjustments. Or even if you're just trying to set it up and you want to make those fine adjustments, you can. It also fits on a CIRS lock bar so that it's easily reproducibly set up every day with respect to your couch. Software like PIPS Pro makes analyzing these images really quick and easy. And of course, you'll need your beam calibration tools periodically as you're doing your monitoring of your beam as well. But the one that I want to focus on for a little bit is actually that I would strongly recommend LinacView, Standard Imaging's LinacView software as well.
And why would you recommend that?
Well, if you're treating stereotactic, you are counting on your machine to have extremely high accuracy in your MLC placement and your angles and your beam delivery. So LinacView is a log file analysis tool, and it can be used to track every single one of these machine settings, not only for calculating the planned fluence versus delivered fluence, say for your pretreatment QA, but you can do the same, that fluence comparison and all of those parameters can be monitored for every single treatment. of every single SRS patient, well, every single patient that you treat. So you can monitor your MLC leaves very closely. You can keep tabs on those gantry angles, the collimator settings, all in really extremely fine detail and completely automated, running in the background. So the nature of these treatments being few fractions, very tight margins, it's really critical that we not just do our pre-treatment QA and then hope the machine's good enough. It's nice to keep tabs on it, to know what it's doing, to know that it's accurate for all of your treatments.
Agreed. It's certainly important.
I think I have a workflow diagram here, just since this isn't as well known a product. A lot of the stuff we've talked about today, other than maybe some of the new phantoms, are things that people have heard about from us for quite a while. How Linac View really fits into your workflow is that once you have a plan generated on your TPS, you would export that plan to your Linac. And yes, this even works with Elekta's Unity, MR Linac. And you would export it to Linac View. Once the Linac View has received that RT plan, any log files that are generated for the patient treatment will be automatically brought into Linac View, associated with the right RT plan, analyzed. Any alerts would show up on screen right there. But then, if something's a little funny, if you have a beam interrupt for whatever reason, or something else, some other alert is generated, you can export today's treatment as a new RT plan that you can re-import into your TPS and calculate the dose with that algorithm that you've commissioned, that you trust, and compare those doses with what you intended to deliver. So it's a really nice kind of security blanket safety mechanism running in the background, making sure that on these highly critical, high dose, few fraction treatments, as well as the rest of your treatments, of course, but focusing on SRS today, you know that your machine is actually delivering what it should and functioning up to par.
That's great. And like you said, it's automated and running in the background, so they don't need to do any specific import in order to get alerts that something is off in the log files. When the patient's treated.
That's
great.
Exactly
we all love automation.
Yes, we do. Yes, we do. So I think that brings us to the end of our QA Essentials talk for SRS and SBRT. Thank you, Andrea, so much for the discussion. This has been really fun.
And thank you. I can't do it alone.
If anybody has questions, please put them into the GoToWebinar dialog box. I'll review those and see if I missed any more. But we'll take a look at that now. But while you're doing that, I would remind you we have a wealth of information about our products and services on our website. And you can always contact one of us directly at the emails that we're showing here. We'll also say, I guess, oh, wait, hang on. Where's my question dialog? There you go.
Oh, did you have it?
No, okay. I have it. Yep. Okay. Are the detectors listed in TRS 483 with correction factors? No, they're not. Actually, I don't know about the D1V and D1H. I'll have to check that. They're a little bit too new. 483, maybe. Now I'm getting mixed up. I was thinking 398. The question was 483. So that's the joint one from the IAEA and the APM on small field dosimetry, I believe. And the W1 is listed in that publication with KQ of 1, Unity in all of those tables. The W2 is identical. The fiber is identical to the W1. And actually, if you buy a W2, you get a package that has both the 1x1 fiber, that is the fine resolution tip, and a 1x3 millimeter tip that exactly matches the W1 fibers. So you can prove to yourself that things are equivalent, but they really are identical fibers. The KQ factors are identical because there's no change in what's being done with those. But I think that was the only other question so far. that we hadn't answered yet.
Great.
So we'll give people another minute or so to add questions if they think of them. Yes, not seeing any questions. I would say we really appreciate everyone's time here today. We thank you for joining us. We hope to see you in the next QA Essentials webinars. And remind you once again, there's a wealth of information on our website, recorded webinars, future webinars, as well as product-specific information, of course, and a way to contact us. So I guess thank you all for taking time out of your busy day. Have a great rest of your day wherever you may be, and we hope to see you again soon in the future.
Absolutely. Take care, everybody.
Bye.
Bye-bye.